So next one is a photophosphorylation by chemiosmotic hypothesis. During the process of electron and non-cyclic electron transport system, here the accumulation of the protons are taking place towards the lumen side from the stroma side, and here this the process was first described by Arnon, but it is in accordance with the chemiosmotic hypothesis, which was proposed by Peter Mitchell. So here the photophosphorylation is taking place. The photophosphorylation means the synthesis of ATP molecules in the presence of light. So this photophosphorylation was first described by Arnon scientist, but this process of photophosphorylation was explained in accordance with the chemiosmotic hypothesis, which was proposed by Peter Mitchell scientist. So here in this process, as we know that cyclic electron transport and the non-cyclic electron transport systems are operated with the help of PS1 and PS2. In this process, in the non-cyclic electron transport, ATP is synthesized. The synthesis of ATP when non-cyclic electron transport is taking place, we call it as a non-cyclic photophosphorylation. And the same way, the ATP synthesis will take place in cyclic electron transport system also. So we call that process of ATP synthesis during cyclic electron transport as a cyclic photophosphorylation. So all this ATP synthesis based on the concentration difference of the proton molecules on either side of thylakoid membrane. So the either side means that is lumen side and stroma side. So this is called as a proton concentration gradient. So the proton concentration gradient is a basis for the process of photophosphorylation, where the synthesis of ATP is possible. So here, as we know that by quinone cycle and by photolysis of water, the protons are accumulated towards the lumen side, and there is a decrease in the number of protons towards the stroma side. So like one is quinone cycle, where from PQH2 the protons are, 2 protons are accumulated, and at the same time the photolysis of water, two protons. So here when one photolysis of water is taking place, the quinone cycle will be operated twice, so that a total of 4 protons are accumulated. So a total of 6 protons are accumulated towards the lumen side, and there is a loss of 4 protons from the stroma side. So here the decrease in the number of protons on stroma and increase in the number of protons on the lumen side is taking place. So because of this one, here there is a concentration gradient of protons is established on either side of the thylakoid membrane, that is lumen and the stroma. So this is the way how as protons are being accumulated. So here the protons will try to move towards the stroma side from lumen, but the thylakoid membrane is impermeable, but there is one passage through which the protons are moving from lumen to the stroma side. So that part is called as a ATP synthase. Or coupling factor. So this is also been present in the thylakoid membrane. In the non-appressed region of the thylakoid membrane, this coupling factor or ATP synthase is present. So if you see the structural representation of this ATP synthase, so it is present in the embedded in the thylakoid membrane. This is the thylakoid membrane, and the two parts are there. One is called as a CF0, that is CF0, and CF1 regions are there. Which is acting as an proton channel. So this forms the thylakoid membrane. So this part is called as a CF1, and this region is called as a CF0. So this part. Here CF0 is acting as an proton channel, and CF is acting as a catalytic site. Here this is the lumen, and this forms the stroma side. In the lumen, the accumulation of protons was taking place, and the proton concentration gradient is established. So here, when 3 protons are moving through this proton channel, and this some amount of energy will be formed. So that energy is useful for the conformational change in the CF1. This conformational change will help in the union of ADP with the inorganic phosphate, leading to the formation of ATP molecule. So ADP will get united with inorganic phosphate to form ATP molecule. So here it is a site where the ATP is synthesized, so we call it as a catalytic site. Likewise, the two parts are present in the proton channel, in which the CF0 is embedded in the thylakoid membrane, and CF1 part is protruded into the stroma side. So here, when 3 protons are moving through the proton channel, that is CF0, so some amount of energy is released, which help in the conformational change in the structure of CF1, and that lead to the formation of ATP molecule. That means when 3 protons are moving through the coupling factor or ATP synthase, so it leads to formation of one ATP molecule. So in the non-cyclic electron transport system, we are seeing that total of 6 protons are accumulated into the lumen side. So when 3 protons are moving, it helps in the formation of one ATP molecule. So as for 6 protons, 2 ATP molecules are formed in non-cyclic electron transport system. And apart from this one, already we have discussed that one NADPH is also been formed. So that means in one non-cyclic electron transport system, because of the chemiosmotic hypothesis or the photophosphorylation by chemiosmotic hypothesis, 2 ATP molecules are being formed. So likewise, the same mechanism will be operated in the cyclic electron transport system, where the concentration gradient is established between lumen and the stroma side, that is leading to the concentration gradient of more number of protons towards the lumen side. They also try to move towards the stroma side. In that process, they will move through the CF0, and that is coupling factor or the ATP synthase, leading to the formation of ATP molecule. So this is the way how this chemiosmotic hypothesis was explained by Peter Mitchell. The same mechanism is operated in the electron transport of mitochondria also. Here the same mechanism is there both in the ATP synthesis of chloroplast and also in the mitochondria, that was been explained by Peter Mitchell. So let us see the reasons for the development of proton gradient on either side of thylakoid membrane. The first one is the splitting of water molecule. when the water molecule splits, so as we know that the oxygen evolving complex which is present towards the lumen side is responsible for splitting of water molecule, where here when the water molecule split, it will forms oxygen, electrons and protons. So these electrons have been accepted by the PS2, but the protons have been released into lumen side. So this is one reason for increasing the number of protons on the lumen side. The second one is the quinone cycle. So mainly that plasto semiquinone. So this is the partially reduced form of quinone, in which it is accepting, when it is taking the electrons, one electron from the, it will take up 2 protons from the stroma side. So this is not only an electron acceptor but also a proton translocator. So this is the one which is, because of which when one quinone cycle is operated, 2 protons have been lost from the stroma and 2 protons have been gained towards the lumen side. Loss of protons on stroma side and gain of protons on lumen side. Here the third one is a NADP reductase enzyme. So here in the final step of non-cyclic electron transport system, the electrons have been accepted by NADP plus, and not only it will accept the electrons but also it will accept protons which are present in the stroma. So that means here loss of protons on stroma side because of NADP reductase enzyme. So like here what is happening means because of some reasons the number of protons are decreasing in the stroma side, and at the same time the gain of protons is taking place on the lumen side. So here because of this one, the concentration of protons is increasing, the proton concentration gradient of proton is increasing, and the pH is decreasing. So here these are the 3 reasons where this reason for chemiosmotic hypothesis, where the protons will get accumulated towards the lumen side and decrease in the number of protons on the stroma side. So here because of all this one, pH is decreasing in lumen side. Finally here this catalytic and the embedded site, that is ATP synthase, is the one which is responsible for diffusion of the protons from high concentrated lumen side to the low concentrated stroma side. So the diffusion is leading here actually the CF0, which is acting as an transportation channel. So when 3 protons are transported through CF0, so it is releasing some energy which is useful for conformational change in the CF1. The CF1, when it undergoes conformational change, so that will be helping in the formation of ATP from ADP and inorganic phosphate. Likewise, the chemiosmotic hypothesis is been explaining the process of ATP formation by photophosphorylation, and for all this the basic reason is a concentration gradient of the protons, or proton concentration gradient. For all this process they require a membrane, that is a thylakoid membrane. Here the proton pump is required to facilitate the diffusion of protons from high concentrated region to the low concentrated region, and proton gradient is also being required. Along with this one, the ATPase. So these are the primary requirements for chemiosmosis. So here in this entire process where ATP synthesis is taking place along with NADPH. So this is a way how this chemiosmotic hypothesis explained is useful to explain the photophosphorylation in the chloroplast leading to the formation of ATP molecules. as a result of non-cyclic and cyclic electron transport systems, that is by non-cyclic photophosphorylation and cyclic photophosphorylation, the energy forms, that is ATP and NADPH are formed. So these are considered to be the assimilatory powers. So these are formed in the thylakoid membrane, or they have been formed released into the stroma side because of the thylakoid membrane, and these ATP which have been formed in the towards the stroma side, and also the NADPH is formed towards the stroma side, which will be utilized in carbon dioxide fixation, where the other substrate molecule of photosynthesis, that is carbon dioxide, get reduced to carbohydrates. So now in this first step of the photosynthesis, that is a light reaction, one of the substrate, that is water molecule, is undergoing oxidation and releasing oxygen. And in the second step, so in the first step, not only releasing oxygen but also synthesis of ATP and NADPH is taking place. The so formed ATP and NADPH2 are utilized in the second step, that is we call it as dark reaction or carbon dioxide fixation, where the carbon dioxide molecule is reduced to carbohydrates. So that will be studied as the C3 cycle in some plants and C4 cycle in some other plants.